David Johns, welcome to the podcast.
Thank you for having me.
It's such a pleasure. You're joining me from the UK. Now, whereabouts in the UK are you?
So I'm in Plymouth, which is in the far southwest of the country. It's lovely at the moment, beautiful weather. Plymouth's not normally like this because we stick out into the Atlantic. We're normally hit by all the bad weather. But yeah, it's beautiful now. So it's a good time of year.
Now, are you originally from Plymouth?
Yes, yeah. I hadn't intended to stay all my working career here, but that's just the way it panned out. You probably can hear my accent. I've got it's a very sort of country accent, I would say. People in the southwest have quite a broad accent.
Okay, so Plymouth, is it more like a very small town?
It's a small city, a very small city. So we're quite away from London. If you wanted to go to London, we're about 4 hours by train. So you can imagine everything pretty much stops maybe 2 hours away from us. There's not much down here.
Okay, very cool. I kind of grew up in Northern Ontario, Canada. I'm not sure if you know our provinces really well, but in a very small town as well. So I can kind of relate to that. So I want to present you to people. You are a plankton ecologist. You're a scientist. You work with plankton. Obviously, we're going to get to that in just a second here. You're also the head of the Continuous Plankton Recorder Survey, which is something I also want to discuss.
Sure.
And just before we got started, I asked you if you wanted to be called Dr. David Johns, and you said, well, I'm not actually a doctor. And so I'm really curious about that because, you have so many credentials to your name, but you don't have a PhD. So why did you stop at, I'm assuming you did a master's?
Yeah, I actually, it's a good question. I went to do a PhD and was doing that quite well. I ended up submitting it early as an MPhil. And really, not to get into the detail, it was at the time my wife and I were fostering one of our niece who had lots of problems, lots of mental health issues. But it basically it just meant I couldn't focus so much really on that area that I wanted to. But when I was chatting at the time to a few people that I work with, they were kind of like, well, the PhD is nice to have and the doctor title is nice to have, but actually having the publication output and the reputation of your work is probably more important. So I kind of came to the conclusion that actually it shouldn't really hold me back. And it doesn't so much in the UK. I know it does in some countries. It's kind of PhD is seen as the badge and you're not going to progress anywhere. But actually in the UK, it's pretty good.
Yeah, and the reason I asked that question is simply because, I do get a percentage of students that listen to this podcast, right? I mean, they want to listen to the scientists that are the leading scientists in their field and stuff like that.
Sure.
So it's always refreshing to get somebody who didn't necessarily become a doctor and yet still does, so much work and has, like you said, all the credentials you need in your field. So that's really exceptional.
Yeah, and it's kind of unusual, but I kind of like that really, because if I attend a meeting or a conference and you'll see doctor, Mister, I always think, I'm like the specialist in the room. If you go to the hospital, they always call themselves Mister. I'm like, yeah, that's me.
That's nice. Yeah, it's funny because I when I was researching your work, I saw the title Mr. David Johns, and I was like, oh, no, that's unusual. I'm going to have to ask him about that. Well, that answers that question then. Okay, so plankton ecologists. Now, I've had somebody that we both know, Dr. Steve Thackeray, on the show already, and he's an ecologist as well. I've had Marianne Denton, who's an ecologist. I've had quite a few ecologists on the show. Probably the most common scientists I've interviewed are ecologists. But specifically, you do plankton ecology. Now, first question on that is it saltwater plankton or freshwater or both?
Yeah, all saltwater, all marine. No freshwater stuff for me.
Okay, that's interesting because Steve's work, if I recall correctly, was freshwater. So this is going to be interesting in terms of the differences here.
Yeah.
Okay. Can you explain to the lay people out there, in your words, what exactly are plankton?
Sure. They are generally, when you're talking about sort of marine plankton, generally microscopic organisms. When I say microscopic, they can actually range up to a few meters across. So a large jellyfish would be thought of as planktonic. but really plankton sort of refers to the organisms that are subject to the sort of the whims of the currents and the tides. They're sort of moved around, and I think plankton comes from the Greek word planktos, which means to drift. So in the marine environment, there are animal plankton, which are zooplankton, and plant plankton, which are phytoplankton. And there's thousands and thousands of different types that we see, but generally the phytoplankton perform much the same way that terrestrial plants perform, and they perform photosynthesis, so therefore they produce oxygen. A lot of people wouldn't know that they actually produce around about 50% of all the world's oxygen come from phytoplankton. And then the animal plankton, the zooplankton, some of them stay in the plankton their whole life. They don't live very long. But some of them develop into sort of the animals that most people are familiar with seeing, sort of like crabs, lobsters, sea urchins, starfish. They all start their life in the plankton as these tiny little microscopic creatures that eventually settle down and then turn into things that we're more familiar with.
Okay, hold on. So a lobster starts as a plankton?
Sure, yeah. They start as
No way.
they start as something that would look nothing like it. Shame I don't have any pictures to show you, but they're like a little weird crustacean, very tiny. A lot of them have very large spines that come off their heads. There's some of them that they would look nothing like a crab or a lobster. You'd have no idea. They're very distinctive when you see them, but then they go through to this stage called a megalopa, where they start to look more like a lobster or a crab. And then when they get past that stage, they settle to the bottom, and then they're a lobster or a crab.
Do you know how long that process takes?
I don't think it's very long. I think it's in the order of months. It's not very long, but they're tiny. We see them in the plankton as these megalopa stage. You could tell something was a crab. So it's got its little pincers. It's got a little tiny tail, but you're talking, 2 millimeters. It's very, very small. And then it would settle down, and then it would start to develop from there.
I thought that smaller lobsters, I thought that lobsters would be like the size of crawfish. I had no idea that it would go from this microscopic thing into a full-fledged lobster.
Yeah, absolutely tiny. I think it takes a long time, doesn't it, for lobsters to grow? It'll take a long, long time. But they all start their life as these tiny little eggs, and then floating around in the plankton and eventually settle down. Yeah, it's very strange when you see some of them. We actually did a game because we do a lot of outreach where it was match the juvenile stage with the adult stage, and for sort of crabs and lobsters and starfish and sea urchins, they just don't look anything. It doesn't look anything like it. It's guesswork for everybody unless you know.
Yeah, I'm definitely going to Google that one after this podcast. What about other creatures? So I know a bit about plankton because I examine creatures under the microscope all the time out of pleasure. There are other crustaceans that pretty much almost remain microscopic, like copepods, or we like to call them cyclops.
Yeah.
And so are they relatives of lobsters and crabs?
Yeah, they're all crustaceans still. So copepods is a good example to sort of talk about, because they're one of the most dominant organisms in the sea, actually. And for us, we'll go on probably to chat more about the CPR survey. We record, 800, 900 different taxa, and most of the work is focused just on the copepods, and probably pretty much just on 2 copepods. That's all people are interested in, because they play such a critical role in the marine ecosystem. But they're very similar to the stuff that you would see, but some of them are just a little bit bigger.
Right. I assume always that the freshwater versions are always smaller than the marine versions. But in the freshwater environment, I've heard that copepods are actually considered a pest. Is that the same in the marine environment?
No, not at all. None of them, as far as I know, none of them would be seen as to be harmful. And in fact, they're keystone species, really. So there's one of them, which you could chat about a little bit, called Calanus, and there's one called Calanus Finmarchicus, and it's probably, as an adult, maybe 4 or 5 millimeters long. It's very small. If you were to look up plankton, probably 90% of all research has been done on this guy, but it plays such a critical role. So larval fish like to eat it, other zooplankton like to eat it, but it's also eaten by cetaceans, whales, dolphins, seabirds will pick them off the surface. The complete opposite of a pest. It's vital. In fact, a lot of the work we do is monitoring the sort of health of those populations, because they have such an impact on the rest of the ecosystem.
You mentioned just right now key species.
Mm-hmm.
So I guess there are several species that you've identified as perhaps more crucial than others. How does that work, and what are other examples of other key species?
Yeah, I suppose we sort of try to think of them as key species. I suppose everything's got a place, isn't it? Really, but these guys, these sort of copepods, certain of the phytoplankton, I would suppose, they play just this pivotal role, and Calanus particularly. That's the one we really know so much about. Everything within a lot of the ecosystems depend on it. It's sort of like I said, it's eaten by fish, eaten by larval fish and by larger fish. And actually, part of the sort of Calanus life cycle is they store fats, lipids in their body, so they act as this link between sort of like the primary producers and then the secondary consumers. And if you didn't have them, you basically wouldn't have a healthy fish population. So in the UK, everybody, you probably know, everybody likes to eat cod. It's cod over here. Everybody likes cod. It's like a very conservative palate. And Calanus are vital for sort of cod larvae development. So anything that's happening to the Calanus has this ultimate impact onto cod populations.
Okay, yeah, I definitely want to get to speaking about their population in just a minute, but I want to stick a little bit to learning more about the plankton in general. And so Calanus is a very important one. You mentioned phytoplankton. What would be one that you're really, really looking at?
The phytoplankton one is we tend to quite lump them in a way because there's lots of species. And when you start to do sort of any sort of population work, they are all doing various different things. We've got long-term data sets, and it can be quite noisy if you look at it. So you tend to group them into like what we call functional groups. So within the phytoplankton, you have mainly diatoms, which are little tiny primary producers, which are like a little silicate shell. You probably get them in freshwater. I know you do.
Oh, definitely.
And some of them are a little round, like little petri dishes, and some of them are long and thin. But the other big group are Dinoflagellates, which are kind of what you call them, kind of mixotrophic, so they can photosynthesize, but they can also ingest things as well. So we tend to quite often lump most of ours into these two groups. So we have a look at what total diatoms are doing and what total dinoflagellates are doing, see how they're changing in relation to each other, because the sort of population of the two together can sort of tell you a little bit about what's going on in the environment.
What about creatures that change with the seasons? So, for example, in freshwater, there's a Sinura. There are Volvox, for example, will change with the seasons. They tend to appear in the colder months, not so much in the warmer months. Is that the case with the oceans? Do we get
Mm-hmm.
creatures that appear based on temperature and seasons?
Sure, yeah, very much so. So sort of particularly around uh, the North Atlantic and the sort of Northeast Atlantic, where we're based, there's very strong seasonality, and the plankton blooms in the springtime, much like you would get you go down your woods and you would see it, and that's kind of a factor of temperature, as you mentioned, but also light levels. It needs a particular amount of light in the daylight to sort of spark them off. And then you get this peak in the spring of diatoms. They're the ones that respond really rapidly. And you tend to get very large populations of them. And at the same time, the sea around the sort of UK starts to become stratified. It warms up, and there's no vertical mixing. So what you get then is the surface nutrients start to get depleted. So these diatoms, which are photosynthetic and they need this stuff, they start to die out. And then you get the dinoflagellate community takes off because they're mixotrophs, so they can sort of photosynthesize, but also they can sort of consume other things. So that's kind of like the normal way it progresses in the plant plankton. And then quite often at the end of summer, as you're going into autumn, we tend to get over here, it becomes windy, stormy, the end of the summer, and mixing occurs again in the oceans, which then resuspends nutrients, and then you get another diatom bloom. So you get diatom bloom in the spring, dinoflagellate bloom through the summer, and then another little diatom bloom in the autumn. And that kind of is the classic way that it acts, and you can see that from the data very clearly. And then there's a number of the sort
Okay.
of animal plankton, the zooplankton will respond along this. Some like to be there right at the front when the diatoms are kicking off. So Calanus, for example, they sort of time their egg productions, whatever, just after the bloom. So there's lots of food then to eat. And then some of the other stuff start to come later in the year. So yeah, very clear seasonality.
I'm just curious, do you ever look at marine tardigrades, or have you seen anyone doing your research?
Never seen any. No, never seen it. And it's
Really?
probably, and I think I know they're tough. I know they're very, very tough. I just wonder whether or not being fixed in formalin, because our samples, we fix them kind of on the fly with formalin. So everything that comes back is really well and truly dead. So I imagine because they go into like what do they call it? A ton stage, the resting stage.
Yeah, like cryptobiosis, like a ton state, yeah.
Yeah, and I wouldn't know what that looked like. So if it didn't look like a little piglet, we probably wouldn't recognize it.
I just always ask, I've interviewed some marine biologists and stuff, and I'm always like, have you run into a marine tardigrade yet? So yeah, it's interesting because my partner and I are actually moving to the East Coast next year. So we're moving to Prince Edward Island here in Canada.
Okay.
And it'll be my first experience with the marine environment. So I'll be sampling all sorts of things, and I'm curious to know, you research, for scientific purposes, but
Mm.
somebody like myself who's not a scientist and moves to a marine environment, would you recommend that I should like sample tide pools to look at plankton? Should I sample the shore? Should I throw a plankton net over a bridge or something? Like,
Yeah,
what's the best way?
I think some of the certain times of the year, I think they probably all got their advantages, to be honest, and you're going to see slightly different communities. The sort of chucking the plankton net in works really well, like surprisingly well. We've had a couple of guys who do that. If we have students coming in to have a look, because all our plankton tends to be dead, we have people just going to the shoreline and just literally throw it off, tow it around, bring it back, and this where we now June to August, it'll be rammed with organisms, absolutely rammed. There'll be so much to see. Lots of jellies. It's what you see a lot of. Lots of little tiny jellies that are difficult to identify.
Yeah. I've never seen a real live jellyfish. I've
You'll
been
get
to
a lot.
Portugal. I saw some dead ones on the beach, but I never saw a real life one.
Oh yeah, you'll definitely see them there.
Oh yeah. Do you know if like really small jellyfish are they poisonous when they're that small,
No,
or?
not at all.
Okay.
No, they'd be far too small. We see them on the samples a lot. The thing is, with the CPR sample, we'll probably go on to explain it. It's towed behind merchant vessels, so it's going pretty quick. It can be doing 20 knots. So when it hits a jellyfish, you can imagine it. But what you see on the sample are the nematocysts, the stinging cells. So they're really clear. You can see them really, really clearly. But they're tiny at that stage. They're really, really tiny. They'd never penetrate your skin. It needs to be a slightly heftier organism.
So what would you say, I guess, for people who are like, well, why are you studying this microscopic life? Like, why does it matter? Like you gave the cod example, which I think is a really convincing one, which is, guys, if we don't study this microscopic life, we're not going to get as much cod, and then, we're not going to have as much fish and chips. So like, what would you say to people, why study the microscopic?
Sure, yeah. Well, there's loads of reasons really, but one of the things you can sort of call it is it's the canary in the coal mine, the plankton community. Anything that is going to happen is going to happen there first. The plankton, they're multi-generational on a small time scale. They react really rapidly. If you think of something like an oak tree, an oak tree, it would take you ages for it to see anything going on, or it would take you ages to see if climate was having an impact, unless it was a massive sudden event. But with the plankton, because they reproduce so rapidly and the community is just turning over, you can see anything going on. So climate impacts, pollution impacts, you can see them straight away. But because they're so important to fisheries, and so they're so important to all marine life, that's the reason we say that you need to study them. They give you this idea of what's going on in the ocean. People like to talk a lot now about this term called multiple stressors. So if you think about the marine life and you think about fish, the main stress on fish in most places is overfishing, isn't it? It's anthropogenic impacts. But that's not the only thing impacting fish. There's also the temperature from climate change. There's also pollution. but there's also the impact on what they eat. So what's going on with the plankton will impact on the fish as well. So it starts to add up this sort of multiple stressors, and you need to get a handle on all that, because you couldn't put any management measures in place without knowing everything. You could say, actually, in the UK, we'll stop fishing for cod, no problem. The stocks will recover. Well, they probably wouldn't recover because it's too warm for them in most of the UK waters, and they're moving north, as well as their plankton food stuff of choice is moving north as well. So there's lots of sort of things to consider. So that's kind of why we say it's important.
So I guess, like in this cod example, the cod would just follow the stuff that they eat, essentially.
Yes, yeah. So it's kind of, they're following that, but it's also they prefer colder water, and their food stuff prefers colder water. So they're both kind of shifting away. And we've seen in sort of our results, we've seen around about a 10-degree shift in latitude over a couple of decades around the UK.
Whoa.
of the plankton community. So you get the colder water, the preferring plankton has shifted northwards, and there's been a shift up from the south of the warmer water plankton, but it's not a like for like replacement. So if you sort of go back to the Calanus I was talking about, so the one that the cod really like is called Calanus Finmarchicus, which is a cold water form. You get it off Canada as well. There's a chance if you sample on the East Coast, you'll see them there. They're very common. But you also get a warm water form called Calanus Helgolandicus. So that's moved up as this other Calanus has moved away. They both co-occur, which is a real pain because they look almost identical. And to separate them in analysis is really tricky. These things are 5 millimeters long, really, really small. And their rear leg, the inside, which would be the inside of their thigh, has these little teeth. So you have to look for these little teeth for the angle they're at. That's the only way really of reliably doing it without using genetic tests. And considering that you get thousands of them in the sample, that it just would be impossible to do. So so the cold water form has moved further north. The warmer water one has moved in. They haven't replaced each number like for like. And on top of that, they also have a slightly different seasonal cycle. So the Helgolandicus, the warm one, tends to peak later in the year, which is not so good for the cod. And they also store lipids. I mentioned about them storing lipids. The Calanus Finmarchicus generally stores lipids more effectively as part of its life cycle, whereas Helgolandicus doesn't. So it's kind of like there's less of them. They peak a different time of the year, and they're potentially less nutritious. So it's quite an impact.
And the cod knows which one is which.
I don't know if they can. If you put it in, they were to choose, I would. Yeah. I don't know.
That would be a fun experiment to do.
It would, and that would be an easy way of identifying.
Yeah, exactly. Do you know if this kind of survey is being done in other countries around the world? Is it really unique to the UK?
No, it is being done in other countries. So it started in the UK. The very first kind of CPR tour was 1931. So it's actually our 90th anniversary this year. And then it's kind of moved around. So we have a survey that we run in the Pacific that's been running for about 20 years. There's a survey that's run in the Gulf of Maine, which was run by the US. The US stopped funding it, but now they're funding it again, but we're running that one. And then there's one in Australia, there's one in New Zealand. There was one in Cyprus, but I think the funding stopped there. Brazil, I think, have been doing some work. So there's a few scattered around, and it's kind of like it's a Global Association of CPR Surveys or something. GACS. It's a horrible acronym.
So then I guess I'm going to have to lobby the scientists in Canada to start one on the East Coast.
Yeah. Well, actually, we do run a route. A number of routes in that area. We have one that runs from Iceland down to the tip of Newfoundland. St. John's, I think it would be. And then we've got another one that runs from the sort of bottom of Newfoundland across to the Gulf of Maine. And those routes are actually partly funded by DFO. So DFO do that now.
No way.
Which is DFO.
That is so cool.
I'm assuming you know DFO, but it's it's the sort of like the department. Is it Fisheries and Oceans? I can't think what it stands for. It's a big.
Yeah, actually, it's funny because my partner works for DFO.
Oh,
so
okay.
it's quite fascinating. I had no idea that they had a partnership with the UK to do that. That is really, really cool.
Yeah. It's good. It's really good. And we actually work with them and with NOAA from the US, and they kind of both part fund a number of routes, and then they share the data. So we're very open with the data, and we let anybody have the data if they want it.
So, okay, so the Continuous Plankton Recorder Survey, let's just call it the CPR because you've been
Yeah.
using that acronym. Okay, so you said it started in 1931. Was there a reason why they started it? Did they really know back then that we this was important to study?
Not really. No, it was started by a guy called Alistair Hardy, who was a fisheries scientist. So until I know anybody listening to this might know, might not know. So the CPR survey was actually called the Sir Alistair Hardy Foundation up until only a few years ago, and then we merged with the MBA. If you can see my T-shirt, so we've now
David's
merged
got
with.
his shirt
Got
on.
my shirt on.
Yeah.
So we merged with them. But yeah, Alistair Hardy was based in Hull, which is sort of the northeast of the UK, and he was in contact with local fishermen, and they'd been noticed. They were herring fishermen actually. They had noticed that when they were going out casting their nets for fish, if the watercolor was a certain color, they'd either get more fish or less fish. So they'd been chatting to him, saying, "Oh, this is really weird. What's going on?" So he designed a plankton indicator, kind of the forerunner of the CPR, which was a very small metal tube, maybe, I don't know, like a foot long. It was like a very mini torpedo. They could throw it off the back, and it had little silk discs which act like a little mesh, and they could bring it back. And if it had plankton on it, he provided instructions. If there were certain plankton on it, like the Calanus, they would know to throw throw their nets overboard. And we actually found recently one of his original adverts, and I'll have to email it to you because you would never get away
Please
with it
do.
now. It's just kind of like, this is guaranteed to increase your catch by 80% a night. And it was just, I always think of him as a little bit like, what's he called? The guy from The Greatest Showman, where you really, really sell.
Yes.
And I think the only way, you think it's been running 90 years, you need somebody like that who's kind of a visionary, but also, can really sell something.
You mentioned watercolor. Do plankton change the color of water?
Sure, yes, they do. Yeah, so you get lots of plankton blooms. Mainly the phytoplankton blooms can change the water, so it can be green, brown, red, depending on the species. And that sometimes is quite a good idea, an indicator of what's in the water. So if it's red, it tends to be a dinoflagellate bloom. We get this thing called Phaeocystus, which is kind of like this disgusting foamy slime, and that makes the water go brown. So interesting. I was talking to somebody today, that potentially another collaborator, about looking at Phaeocystus, and it's in the UK, and it's probably the same in most countries. People report it as sewage. They say it's sewage outflow, but it's not. It's actually a phytoplankton, and it just blooms, and then the wave action just makes it froth up, and it looks foul. It looks really horrible. So if you see that in the water, it's
Wow.
normally not sewage. It's fine to swim in. It would just feel disgusting.
It's interesting because I recently took some samples about a month ago in a very small temporary pond,
Mhm.
and now I don't know if there's a marine equivalent to this creature. It's called a euglena. Are there marine equivalents? Do you
So
know
that's
euglena?
yeah, I think so. That you have to remind me what it looks like. Isn't it like a little kind of a little wormy thing, isn't it? That's
Yeah, but it has like
my
a big
scientific.
it's a flagellate, so it has like a flagellate at the end and like an eye at the other end. And anyway, what's interesting about this is that the one I saw was red, and
Yeah.
it turns out that they can become red in response to the UV light.
Oh,
And if
okay.
you have a whole bunch of them, it'll turn the water red, or
Yeah.
it'll appear red.
Yeah.
And so that was my first real encounter with a creature that can change based on environment, you know?
Yeah, yeah, yeah. And I think that's the same with some of the dinophyces as well. I think because, like I said, they're mixotroph, they can absorb other little organisms. They can take in other types of sort of really small algae, and the color of them can change. And then, yeah, you get this bloom. And some of the blooms can be quite toxic. Some of these can cause red tides. If you sort of Google red tide and have a look, they can cause fish kills, and they can actually even give off a gas that can get into the air and can be toxic to people. They can be pretty nasty, to be honest. I think
Yeah,
actually
I don't
some
know
of
if
these.
you've heard of I was just about to say, I don't know if you've heard of the mysterious brain disease crippling New Brunswick, Canada right now. They don't know what it is, but they are suspecting maybe like an algae or something.
Oh, right. Okay. Yeah. Because I know a
Yeah.
lot of the East Coast fisheries gets closed quite a lot through these sort of
Yes.
harmful bloom outbreaks. Yeah. So
Yeah.
it's
So
really
it's
difficult to. Yeah. I was chatting to somebody today about how she was looking at satellite data, and trying to match up satellite data with potential harmful algal blooms, because they look at the like the sort of different light spectrum and how it bounces off from the satellite image. But a lot of these phytoplankton species that can be harmful, they're not harmful all the time, and often we don't know what makes them go harmful. Sometimes it's the sheer volume of them makes them kind of toxic, but other times you can get masses and masses of them and they cause no issue at all. So it's really difficult to see. You could take your water sample and say, oh look, there's like 1 million pseudonychia, it's one of the species in there. That should probably make me really ill, but actually it might not do anything, or it might make you really ill. You don't know, and you'd have to actually go in the genetic route to identify to see which strain you're in there. So it becomes really tricky.
Yeah. So when you're evaluating this through the survey, are you making an urgent note about potential harmful blooms when you survey this, or?
No, we don't. The way that we work, because we've got such a massive survey that sort of covers most of the North Atlantic and most of the Pacific, we're always behind with the analysis of the samples. So we don't do that kind of immediate response. And actually, that was part of my discussion today with somebody. I was saying we were kind of wary of doing that, because it's, you could end up closing an oyster fishery down because you find this particular species, and actually it might be fine. And then, you potentially have lost those people thousands and thousands of dollars. So you have to be really sure,
Okay.
really sure what you're doing.
Yeah, I would imagine those would be like smaller operations, like micro operations that are monitoring this continuously. I would imagine.
Yeah, I should imagine so. I'm not certain what it would be like on your side of the Atlantic, but over here we have it's called the Environment Agency, which is kind of a UK wide, and they go and take samples all the time from close inshore, just all the time. So they take samples of the plankton, of the phytoplankton, to have a look, but they also take samples to look at the chemicals in the water to see if there's toxins present.
Okay, so this CPR survey is essentially, describe to me how it happens. Is it like once a year you guys all hop into a boat, you drag a big giant plankton net around, the ocean? How does it work? How often do you actually survey the water? And how long does it take to process the data? Is it like an annual report that you process? Tell me more.
Sure, yeah. So it's very different to that. So we use ships of opportunity, so merchant vessels, ferries, tankers, all sorts that use their normal routes. So you could have one going from, like I said, one goes from Iceland and it goes across to Newfoundland, and they do that route, transporting cargo maybe every month. And what we do is we ask them to tow the CPR for us. So the CPR is, let me think, it's around about 1 meter, maybe 3 feet long, like a big torpedo. And it's attached to a wire, and they throw it off the back of the vessel whilst the vessel's underway. They don't need to stop. The whole idea is we want to have minimum interruption to those guys, because obviously, that's their livelihood. They're commercial companies. We hardly pay them any money at all to do this. They basically do it as a favor to us. So you couldn't run our survey without the merchant shipping help. And they tow that CPR on their normal monthly route. They can tow the CPR for 450 nautical miles before they have to pull it in. And they can pull it in and they can put a new, what we call an internal, like a cartridge in it. Then they can throw it back over. So if they're going from, say, Iceland to Newfoundland, they'll do that 3 times. They have 3 internals that go in there. And then they get to their port, wherever they're going, and then they will box up the CPR and send it back to us in Plymouth. And it comes back, and then we basically take the sample out. The CPR works by collecting plankton on silk. So we actually use silk mesh. It's pretty difficult to describe like verbally. It's almost like it's like a long sandwich. There's 2 pieces of silk, which are around about 4 inches across, but they're, I don't know, 20, 30 feet long. So 2 long ribbons. They're collected inside the CPR. They're wound up, and they basically unravel as the CPR goes by the action of the propeller on the back, which works a little gearbox, and it advances it. So at any one time, there's a section of silk that's maybe 4 inches by 3 inches exposed in the CPR, which acts as this fishing net, basically. So the plankton gets stuck on it. Then it's advanced by the action of the propeller, and it goes into a storage tank which has formaldehyde in it, which then fixes it underway. So it's all flushed.
It does all the fixing as it's underwater?
Yeah, as it's underwater. There's like a
Wow.
little chamber in the back and some cotton wool, and we put some formaldehyde in there. And as it goes in the water and the seawater sloshing around, it's sufficient to biologically fix it. When it comes back, it gets some sort of secondary fixing as well. But yeah, we've got samples going back to the 50s that in storage that they're okay. You can get some things off them. They're fixed enough. Pretty incredible.
Who designed this contraption?
Yeah, so it was the guy I mentioned earlier, Alistair Hardy. It was kind of like the second or third version that he came up with. They sort of escalated in size from a foot to eventually 3 foot. And the design is virtually unchanged since then. It's all mechanical. It's almost like a clockwork spring inside it. And the idea, the fact that it is mechanical, it doesn't need any power source. You don't need a powered cable, and you can throw it off the back of a tanker at 20 knots and it's fine, which you can't do with most modern pieces of equipment.
Wow. Yeah, that's really, really impressive. As somebody who's very familiar with this contraption, I'm just curious, would you make any improvements to it if you could?
Yeah, yes. I think the way it's made and the metal and everything's perfect. It's nice and solid. But we are thinking of things that we can bolt onto it to give us additional information. We have actually got a project that's just kicking off now where we're gonna potentially put on a holographic imager. So it'll actually take images underwater using lasers that will give you this holographic 3D image of things. So it'll be another way of identifying. So which is crazy. That's going to be trialed
My
in the next
mind is just blown.
next month or so. And you can also put temperature sensors, depth sensors, sort of chlorophyll level sensors, all sorts of things. So you can kind of augment this CPR with that.
Yeah, because these merchant ships, are they sending you data, as a companion to the actual machine? Like, are they letting you know, okay, we dropped it off at this coordinate, this is how deep it was, this was the temperature that day, etc.?
Yeah, they let us know the coordinates, or what we call the shoot and haul coordinates. So they'll say, okay, at 5 p.m. on Monday, we threw it off at such and such, and any sort of course changes, they will also give us the information. So that's kind of how traditionally it's been done. So what we want to move away from is that's good for the sort of the scale the CPR works at. We say it's broad scale. It can sort of it can give you information on large bodies of water. But if you want to look at something we would call fronts, you would get them in fresh water where you get a difference of water masses, different temperature water masses, or eddies, which can be like large scale up to say 100 kilometer, 60 miles or so of different temperature water within the sea. The CPR is kind of okay for that, but it would be better if we could get more accurate positioning. So we're thinking of different ways. You could obviously get GPS. You could ask the GPS from the vessels. But what we would do, like I say, we're really conscious. We don't want to give them any more work. We want sort of like the minimum impact on everything that they do.
What's the deepest that you can go with this survey?
It tows at pretty much a fixed depth. So they let out 25 meters of wire or so, maybe a little bit more at the back, and it flies underwater at about 9 meters. So it's very surface. Now, bearing in mind that we operate behind large vessels with big propellers, we kind of think that what they call the prop wash, the mixing, we're probably sampling the top 25 meters of the sea. So it never goes any deeper than that. You could do it. You could probably do it off a research vessel, but it would be difficult because you'd have to pay out a lot of steel wire and tow it at depth. And, you don't want to bang into anything. We've lost a few where they towed them in too shallow water, or they hit icebergs or whatever. And they're expensive to replace.
Yeah, I bet. Is there anybody who's actually studying like really deep microscopic life and plankton? Because I'm assuming that there are plankton in the deeper end of the ocean, right?
Yes, yeah, very much so. So yeah, they do all different methods. They do sort of like, we think there's different types of nets, different types of devices that you can drop down that will open at sort of timed intervals, so they can go down to thousands of meters and they'll take a sample and go whatever. So lots of guys are doing that stuff. They're really interesting. What they don't have this is me selling the CPR survey, they don't have the spatial scale. So they're very good if you're behind, a scientific cruise ship that you go out on and you say, okay, we're going to sample at this point, 5,000 meters. Let's drop a sample and let's see what's going on. That just gives you that snapshot in time of what's going on in one little area, and that's very expensive to do. And actually, it doesn't give you any sort of spatial information at all. So it's interesting, and there's lots of science you can do with it. You just couldn't do the science that we do.
Okay. So now you have all this data gathered from these cool ships that have done this for you. What do you do with that data? What exactly are you looking for?
So I suppose what we're really about is long-term trends and long-term changes. So we have a consistent data series going back to 1958. So it's one of the longest running. In fact, probably a couple of months ago, we just got a Guinness World Record for the longest running, most spatially extensive data set, which is pretty cool because we covered 7 million miles now we've done, which is, I think, something like 14 times to the moon and back, or some crazy figure.
Wow.
So we have all this data, and it's consistent. So we've used the same methodology, the same instrument. The microscopes that we use, although they're modern, are based on the original versions. The type of analysis we do is the same. We've got standardized procedures, standardized quality assurance. So you can basically say the data that somebody is generating today, that they're making in the lab today, is comparable to what they were doing in 1958. There'll be no difference. Which means when you start to look at the time series of the data, any changes we see, we know it's nothing to do with anything that we've changed. So these are real time changes, real changes through time. So a lot of the work we're doing is we're looking at what's going on in the community. What's changing? How are they changing? How are they changing in relationship to each other? How are they changing with other sort of environmental impacts, sort of like the temperature of the sea, or what's the other thing people think about, sort of like ocean acidification. You've probably heard of that term, how much carbon dioxide there is in the sea, all these sort of things. So we're trying to sort of like have a look to see how things change, why they change, and over time and space. That's something that you can do with our data that you can't do with a lot of these other data sets.
I'm curious about the standardization part. What about things like user error, as we would say in the software world, on the part of the people who are actually throwing the survey out, like the merchant ships and stuff like that? How much of that is taken into consideration? Is there room for error there or not?
Not so much, because the way that the CPR kind of works, they generally either work or they don't work. And the success rate of them is in the high 90%. They're really robust pieces of equipment. I've got friends who've been out into Antarctica and been on surveys and said the only thing that worked consistent was CPR, because it's like 70-year-old technology. There's nothing to go wrong.
Right.
So the guys who throw it over this board, it's very rare that anything goes. Occasionally you get a jam in the system. Occasionally, it might hit a log or something and it bends part of the CPR and it doesn't tow properly. But normally we can get something out of it. And then when it comes
really
back, we have various sort of quality control, quality assurances, procedures in place. So we have a team of analysts who are looking at the samples. They're all following, guidance of how to do things. And at most stages, their work's kind of checked. It'll be checked by a senior member of the team. They'll be looking out for anomalies in things that they recorded. And then they'll go back and they'll check the sample again. And it's also checked again before it's sort of what we call finalized, before it goes into our database. So there's lots of checks in place. And the team themselves have continuous sort of training.
Right. It sounds like it's just extremely dependable data, essentially.
Yeah.
So what is the thing that's surprised you the most? Like over all the years that you've been working on this project, what's the most surprising finding that you've seen so far?
Oh goodness, there's so many things. I haven't over the last year, unfortunately, not been in the lab very much, but I was prior to that, looking at the microscope most days, and you can see something that you know yourself when you look at these sort of little organisms. I've been doing this job for 24 years, I think it is now. I could look down the microscope and see something that I've seen probably 10,000 times, but it'll be a really nice view of it, or it'll be in an unusual situation. And I'll be thinking, oh my God, this is like amazing. it's like I'm so excited. I've never seen it before, but I have seen it thousands of times. So that's always one nice part of the job. But I suppose some of the results we've seen, some of them they're really interesting scientifically, but they're also quite scary, the sort of impact.
Like what?
So we had a diatom, like a phytoplankton, that we found in the Northwest Atlantic. So it would have been, I'm trying to think where it would be. I've got a little map here. I'm trying to see where the map would be. So it would be probably just to the east of Newfoundland. So around Nova Scotia. So we picked this up on our samples. Very excited. It's totally new. It looks like a little dinky little domino, little dots on it. Really easy to see. And we recognized it because we'd been finding it in our samples in the Pacific. So I got in contact with some guys from DFO and said, have you been seeing this as well? And they had. And it is a Pacific species. So we're finding it in the North Atlantic. And we put a paper out on it. And the hypothesis is, because of reduced ice cover, it's come around the top. It's come around the top from the Pacific into the Atlantic. So it's one of these transoceanic migrations. And we made sure we checked everything to say that it was not ballast water. I don't know if you know that ships at sea, when they've sort of dumped cargo or fuel, they have to take in water to sort of balance themselves, and it's called ballast water. So there's lots of conventions in place that you can't just take ballast water from somewhere and dump it somewhere else. And Canada particularly are really hot on this. They don't allow it at all, ballast water. And the only ships operating in that area were kind of icebreakers and Coast Guard guys from Canada. And they're not even going the whole way around. They were just on the coast. So we're pretty certain that this has definitely come through from reduced ice. So part of you're really excited. Oh my God, this is like amazing discovery. And we're getting it in the sort of scientific literature and getting really excited. And then part of you thinks, oh my God, this is kind of disastrous. What else is going to happen?
Do you remember what year you discovered that?
Yeah, that was in the early 2000s. Wow. And it's still there now. It's not harmful, but it's just the fact that and it was actually in the Atlantic. It's in the sediment records from around about 1 million years ago. So they have found it in deep sediments. So it was there, but you're just thinking, okay, what else is now likely to come through? There could be other things coming through. And what have we done, to the planet that something like that happens?
Yeah, that's pretty alarming. One thing I did notice is that there was a Guardian article, I guess a few years ago, maybe.
Mm-hmm.
I'm not sure when it was published, but I did look it up, and I see that they have a link. So I'm going to put a link actually to the CPR survey, because it is available to the public.
Yeah.
Right. The results of your survey.
Sure. Yeah. It's available to everybody. We have, if people wanted specific data sets or to ask specific questions, they would generally email myself, and then I would kind of help them out or whatever. But we do have a completely open, free data set, which I could send you the link to that one. And that data set goes from 1958 to 2018. So it's one of the largest sort of marine data sets you can get hold of. It has phytoplankton, zooplankton, and everything in there has been consistently counted from 1958 to 2018. So it's a complete time series of this stuff. Totally free. Anybody can have it.
Yeah, I might actually access that. I'm learning how to
play with it.
I'm learning more programming, so I'm just kind of having fun with my own personal projects. But that could be fun to kind of play with that data set. That would be a lot of fun. We have about 10 minutes left. I just want to ask you a little bit about science in the UK. Is it something that is scientific research in the United Kingdom? Is that something that's really well funded? Do you see any flaws with it? Do you see any benefits? What's your overall impression about science in the UK?
Yeah, I think it's pretty well funded. Certainly yes, if I say, oh yeah, it's really well funded in marine science, people would say, no, it's not at all. But it's difficult. I think it's probably like everywhere. It's difficult to get funding a lot of the time. And it's difficult for running something like a survey like ours, a long-term survey. That's quite difficult sometimes to sell to the funders. Some of them will see the benefits. We probably haven't got time to talk about. We give a lot of our data and do a lot of our data into sort of things that are applicable to marine policy. So marine law policy, looking after the ocean. So we can sell that. We've got this data set. Look, it fits exactly with this, and it tells you about the health of the oceans. That's great. Other funders, particularly sort of the pure science, or what they call blue sky science, they just see us and say, this is old fashioned. It's old tech. You've been doing it for 70 years. Who's interested? We need to move on to other things. We need to move on to, I don't know if you know things called gliders, which are like these automatic submarines, like mini submarines. So everybody wants to use gliders, and gliders cost millions of pounds. It's mental. Or they want to use something else or some other new tech. So you've always got this kind of fight. So the CPR survey, as it's developed, we've very much tried to apply our work and apply our science to sort of societal needs. So it could be fish stocks, it could be marine pollution, it could be ecosystem health. There's all these terms like goods and services and blue, whatever they call it, blue carbon. So all the time we're trying to say, look, you can use it for this, you can use it for that. So just to keep it relevant. So as long as you can keep doing that, it's not so bad, but it just means that there's a lot of work is taking up in trying to sort of spin up those proposals and contracts. What you want to say, look, this is a fantastic survey. It does some things well, it does other things not so well. We'll provide all the data to anybody who wants it. Therefore, just give us some money and that's all we'll do. We'll just run this. We're not looking to make a profit or a charity. We'll just do it. But actually, it's never that simple. So you're always working from 3-year, 5-year, 2-year contracts, writing a report, putting in the next one to get the next amount of money. But I imagine that's the same. I know it's the same everywhere.
I was going to say, yeah, that sounds exactly like what I hear from scientists in Canada, where, whether it's agriculture or space research, it's always the same. It's always like in a 3 to 5 year scope. And, you do what you can. With the few minutes that we have left, David, I want to ask you about yourself as a, yes, you're a scientist, but also you're somebody who has other interests. Through the video that we're seeing each other on video. This is only going to be released on audio, but I see this beautiful picture of a kingfisher. And then you told me that you're really into natural history in general. Is that your main interest outside of work, or do you have other, hobbies? Are you an artist? Are you, what else do you do?
Yeah, so natural history, definitely. Where I live in Plymouth, right on the north of Plymouth, there's lots of countryside nearby. So there's lots and lots of wildlife. We get lots of deer just outside the house all the time, foxes and badgers and whatever. So I really like that, and I like looking at insects and spiders. I'm not an expert on any of this. I'm just fascinated with it, and I suppose I like being outdoors. I like cycling. I was doing a lot of cycling, but last year I broke both my arms crashing my bike, so it kind of put me off for a little while. But yeah, anything sort of active. That's me, really. I like to keep busy. I like to be doing stuff. So I like to go to the gym. I like going swimming. Did used to do a lot of running. So anything.
Is there like a period? Is there like a period in history that you wish you could travel back to just for a week or so to kind of study something about it?
Well, that's an interesting question.
Would you go to Easter Island or something?
Yeah, that would be cool. That'd be really cool just to see what happened. Yeah. Yeah, or to see how Stonehenge was built.
Yeah.
Yeah, that would be a good one.
That's true.
Or the pyramids. Yeah, anything like that. I'm not. Yeah. I'm kind of interested in some parts of history, but some people are really, really, really into history. That was never my strong point at school, and I suppose my history knowledge is probably quite shocking. I should imagine.
But I guess just out of fascination, because it sounds like you're a lot like me. You just like you take interest in the natural world around you. Whether, for you, it's also professional, but it's also out of pleasure.
Yeah, definitely. Yeah, I think going out and observing anything. You sit in your garden, sit in the woods, sit outside, or even in the middle of a city. You can see so much. It's just so much wildlife around, or so many different things. And I think it's probably a cliche to say it that the fast society that we've got, that people don't tend to do this. But I got to London every night, and it hasn't been recently because of COVID, but there's so much wildlife in London, and you can just stop and listen. And there's so many different bird calls. There's all sorts. And you think, oh, I can hear this, and I can hear this, and I'll be saying to my colleagues, oh, there's a wren over there. Can you hear that wren? The wren is a really small little bird, and they are unbelievably loud. I don't know if you get them where you are, but like crazy loud for something that's the size of, I don't know, like inch and a half. You think how big its little lungs must be. They are so noisy. And I'm like, oh, you can hear a little male wren over there. And some of my friends are like, what? What are you talking about? I'm like, you do not notice these things. You miss out, don't you? There's so much to sort of take in.
Yeah, the fast-paced lifestyle certainly does that. So on that note, let's remind people, just stop walking where you are, take a look down, take a look to your left, take a look
Yeah.
to your right, and look up and see what's around you. Just take a deep breath and just enjoy. Like you said, even if you're in a city, there's
Hmm.
stuff living in the cracks of the sidewalk. So I think it's a good reminder to share with people. So, David, listen, thanks again for accepting to come on the show. It's been an absolute pleasure. As always, I always feel like, especially with scientists and even with artists, I always feel like there's not enough time in an hour sometimes to
No.
cover a topic. But I think we at least we've educated people as to what the CPR survey is. People can go
Mhm.
look it up. We're going to put a link to the survey. We're going to maybe put a link to the data set if you can
Sure.
send me that. And we'll go from there. But thanks again for coming on the show.
Well, thank you very much for inviting me. It's very interesting. It's a good chat.